One of the things maybe to point out about a TSI test at this point is that, again, there's two different types of sugars in the tubes that we're going to be using. There's glucose and lactose. And you can tell that a sugar is metabolized because acids are produced, which in turn produces a yellow color. So what we're trying to say here is that whenever we see yellow color, that doesn't specify which of the sugars are being produced. All it tells you is that, it doesn't specify which of the sugars are being metabolized. All yellow tells you is that a sugar is being metabolized. And so the way you'd be able to tell exactly which sugar is being metabolized is basically you'd have to hypothesize based on the length of the incubation. So if you are really interested in seeing which specific sugar is being metabolized by a bacterium, probably the TSI test is not the best way to do so. The best way to do so would be to specifically use one of the sugar Durham tubes that we used in a previous lab. Right, so for example, if you want to know if a bacterium can metabolize glucose specifically, then probably it's not a good idea to use the TSI test. It's a little bit too general. Instead, use the glucose sugar tube, the Durham tube. All right. So again, the TSI test, it tells you many different things about a bacterial cell. Not only does it tell you if sugars and proteins can be metabolized, it also tells you if a bacterium is able to metabolize sodium thiosulfate. Right? So that's another way to characterize bacteria. And so the way you'd be able to tell if the bacteria in this tube can metabolize sodium thiosulfate, and there's sodium thiosulfate that's included in the medium for it to eat, if it can eat it. The way you can tell if it's being metabolized is, it turns out that there's one of the products of sodium thiosulfate metabolism is hydrogen sulfide. Right? And so if there's hydrogen sulfide that's being released as a result of sodium thiosulfate metabolism, you'd be able to visualize that because there's something else that's added to the tube that basically acts as a hydrogen sulfide detector. And that thing that acts as a hydrogen sulfide detector is ferrous sulfate. Right? So there's some of this stuff also included in a TSI medium. And so the reason why I consider this to be a hydrogen sulfide detector is because if it gets into contact with any hydrogen sulfide, it ends up producing a black color. So if you inoculate a TSI test and you incubate it and you see something like this happen, for example, again, black just means that there's a hydrogen sulfide in the tube, which indicates that sodium thiosulfate was metabolized. And yellow tells you that one of the sugars was metabolized, right, because acids are produced. If you see something like this, again, as a review at this point, black means that sodium thiosulfate was metabolized. Deep red tells you that there's a basic environment up here, which indicates that one of the proteins are metabolized. Right, so again, the TSI test tells you a few different things about a particular type of bacterium. It tells you if they can metabolize either one of these sugars, and it also tells you if the bacterium can metabolize sodium thiosulfate.